It's a real pleasure for me today to introduce our main speaker. It's Dr. Rama Rao Satyawada. Dr. Satyawada is here for a 3-month sabbatical in which we're comparing notes, if you will, in terms of cytogenetics, and then also exposing him to some of our molecular marker and fish technology. And so we think he's having a good time while he's here, and we hope to send him back with additional information and things that he can use back home. We had an opportunity that opened up just recently, and so the preparations for today were done on fairly short order, and our approach is going to be that Dr. Satyawada has the floor for as long as he wants, and if he uses the whole hour, that's great, because we want to take advantage of his presence while he's here. And for whatever time is left over, I'll chime in at the end. But I'm here all the time, so that's not such a big deal. So a little bit about Dr. Satyawada. So he works up in the northeastern part of India, which is right above Bangladesh. And so that's not an area that you and I think about a lot. But it's certainly an interesting part of the world. He has a few photos that he's going to show us, so you get an idea of the lay of the land in that area. But anyway, he's been there with the Department of Botany for 7 or 8 years approximately, prior to which, and during that time, he served as the department head and stepped down about a year ago and is getting back to full-time science and teaching and so on. And prior to that, he spent his career at Jodhpur, which is in Rajasthan, over near the Pakistan side of India. So he's changed geography and climates and everything else over the course of his career. Anyway, so he's a cytogeneticist, and he's been involved with a few interesting projects where they're trying to take advantage of germplasm for breeding programs, and so he's going to describe to you some of their efforts in that way. So, Dr. Satyawada.
Thank you, David, for the kind words. Good afternoon, everybody. Today, this afternoon, I stand before you representing Northeastern University, Shillong, and my affiliation is with the Department of Biotechnology and Bioinformatics. And before I get into my technical part of the lecture, I thought it would be right for me to talk about the place and the institution which might be interesting. Shillong is the state of Meghalaya is in the northeastern parts of India. Here it is a small state, and we are bordered on the southern side by the Bangladesh, as Professor David has said, and on the eastern side we have our neighbors China and Myanmar. And Shillong is not a very big city, a small city with a small airport, small aircrafts carrying the people from the mainland. But the city itself is expanded over several hills on the region. It's not just on the one hill. So we have a lot of diversity within the state of Meghalaya, and the state is a little bit well developed among all the northeastern states, well connected roads. And the term Meghalaya literally means abode of clouds. So literally we stay most of the year among the clouds, and so the hallmark of a Shillongite is a umbrella in the hand whenever we go for explorations, right? But the region is very beautiful. As you can see, it is full of mountains, hills, and valleys, and a lot of tourists and visitors from all over the India and the world, they make it to the hill station because of the wonderful waterfalls that we have in various numbers, various sizes, various gusts. These are the rapids. We have all type of diversity among the waterfalls. And another interesting thing about the state is that we have a what we call the root bridge thing, where the main the taproot and its associated roots form almost a walkover over the small streams. These are very common there, and people take fascination for such natural beauties of the state. And lately, lot of exotic plantation is also being attempted to augment the loss of the some plantation that has been lost. But it is full of endogenous plants also, like wild bananas and orchids, etc. And with beautiful flowers, also seen with varied colors, varied sizes, varied type of botanical interest. There are certain flowers which are pollinated by a particular type of insect which is available only in that region, so you cannot breed them outside the region. So such interesting things are there which I am not going to dwell. And just about my institution, before that, the city of Shillong is also vibrant with the culture and the religion. We have some beautiful churches in the city, which are most beautiful than the famous Goa churches. And the institution is slightly, oh, sorry. Okay.
That one?
1, 2. All right. So the institution is also relatively a newly established university where I work, with comparatively new buildings but very good facilities of libraries, etc. And it is in this department, biotechnology and bioinformatics, I have been working for the last 7-8 years, as Professor David has mentioned. Now, today the purpose why I am before you is just to share some experiences of our work, which is mainly related to the cytogenetics and also augmented with molecular approaches. A strategy which we have adopted to characterize the germplasm of indigenous germplasm collections. I'll be taking just 2 examples. Vigna, which is a common pulse, which is a full source of protein, and another one is a vegetable okra, which everybody might be familiar with. And these 2 are contrasting examples where we got totally different type of outcome from our approaches. The purpose of sharing this information is to tell you that even today, the classical cytogenetics, when it is augmented with molecular approaches, definitely solves some of the problems. And my colleague, my friend Dr. David would be adding something about cotton, about his own experiences. So first I would like to go for the first example. You see, the present day major problem of the world is increasing population every day. So we have more mouths to feed, that means more food to be produced, and that means the job of a plant breeder is never ending but ever increasing as far as ensuring that there is enough food for everybody. And also there is lately a demand for plant-based products and proteins, especially for food materials. Might be the reason is awareness that perhaps plants can give better protein, better food materials as compared to animal source, although neither of them can be ignored. So, and it is in this context, proteins and the type of quality and variety of proteins that these plants which we have got Vignas they provide would be of interest. Therefore, we have a responsibility for a breeder to provide the breeder with the breeding materials. That is the job, so that we can produce more food and produce the enough materials. And where from these breeding materials comes from? It is not that breeder can obtain from somewhere and somewhere. It is basically either from the natural plant genetic resources available in the nature, or we have to modify them so that these genetic resources variability can be induced either by genetic manipulations or by cytogenetic manipulations or whatever appropriate. So that means here I would like to add that in this context of providing quality materials for plant breeder, a cytogeneticist has an important role to play. And because he is the one who can give materials, breeding materials of both the types. He can identify some of these materials from the nature, or he can help in modifying them by characterizing and by using, adopting appropriate procedures. There is a but. Lately, and then this is not a new thing that I am talking. I mean, this is not any new phenomenon we are mentioning. It has been going on right from the days of Sutton and Boveri proposed the chromosomal theory of inheritance, then followed by Barbara McClintock's classical work of development, cytogenetic stocks, etc. in maize, identification of chromosomes using pectin analysis, Sears, who has developed complete monosomic series, rice. Every plant material has been benefited by the cytogenetical manipulations. Lately, the inputs have not been so far, so it is in this context what we are doing in Nehu, Shillong, is to augment the requirement of breeders. Therefore, what I feel is that as today we have the modern molecular approaches available, we can make better contributions. Therefore, we can think from in a different angle, not from the conventional cytogenetics, but also adopting molecular approaches, combining both and providing the desired materials. So the techniques keep changing and they're getting updated, but the genetic concepts, they remain same. Therefore, today it is time that we should both squash, I mean to say cytogenetical squashes, and also crush plant materials for studying the DNA and molecular thing. There has to be a complementation of both the important areas because it can, if we amalgamate our strengths of conventional cytogenetics with molecular approaches, we would be able to provide more diversity with the genetic stock of materials to be provided to the plant breeders. So this is what we are doing at Shillong. We are using the conventional cytogenetics, and wherever we need, we are confirming or verifying using adopting certain molecular approaches, whether they are molecular markers or there are gene sequencing technologies or transcriptome analysis, whatever it is, we are using those methods and approaches to verify some of our results that we are getting. So this is the objective is finally to provide the breeders who are associated with us so that they can go for genetic improvement and provide better food materials. A major project has been given to us in 2008, which is about to be completed in just in few months, next month is going to be over, with a major objective of working on 3 plants, but I will be covering only Vigna and Abelmouscus, because these are the contrasting examples as I will demonstrate here. And ultimately, even to establish the molecular basis for such diversity, to define it, to establish it, and then adopt it. This is the strategy that has been suggested to us, and we have adopted. Vigna, it is a famous, it is a popular pulse crop, especially those areas where plant proteins are preferred more, and its 2n number is equal to 22, and the genome size is around 579 base pairs. And if you look at this content, protein content, the variability is from 18 to 22% in the seeds. So it is an important source of food material for most of the people who consume these plant proteins, especially countries, Asiatic countries like India, Pakistan, Bangladesh, etc. So it is it comprises of several important plant species, about 170 of them, which includes green gram, black gram, cowpea. Professor B.B. Singh, who is world authority on this, he is with us. He would agree with us that this is one plant which has revolutionized the food requirements. And then we have some underworked or underexplored pulses available in Indian subcontinent. So these pulses are very, very important, especially for Asiatic countries like India, Pakistan, Bangladesh, where vegetarianism is a way of life. Many people prefer plant proteins. So I would demonstrate the diversity in few slides, that which has in not only in the seed color, in the protein content, even in the amino acid profiles of these proteins. Lately there is a report that there are some unique accessions of each. Some of these species, like wild species, like Trinervia umbellata, Trinervia bornea. These are some of the ones which have some unique amino acid combinations in the proteins. And so you can see that the seeds are quite diverse and these things. And what we have done, what we were supposed to do was to acquire more germplasm, to see into the diversity and identify novel genotypes, so that our breeding group will take up them for betterment. So as a cytogeneticist, I viewed from the point of view of this. Sorry. Sorry. Go back.
You want to go backwards? You just use the allowance.
One slide, please. One slide.
Am I going the wrong way?
I have run through some of them.
You want to go this way?
Oh yeah, that's it.
That's the wrong way.
Thank you. So what we have done is about 16 species have been explored and about 90 accessions have been collected, whose details, some of them are cultivated, some of them are wild, some of them are both wild and cultivated. There is a lot of diversity. So these are the materials. So this was our basic approach, that is to start with a cytogenetic approach, identify the problems. If we can solve it, it's fine. If that is the final word, it's okay. But when there are questions, we have shifted to molecular approaches, where we have used mostly the single primer amplification reaction, SPAR approach, where a number of markers have been concatenated into one and then analyze the diversity so that the correct genotypes can be identified. In addition to that, we have a breeding program for which phylogenetic relationships need to be established, especially in view of several new species which we have come across while during our studies, exploration. Not many, but 3-4 of them have come out with unique and novel characters. So how they are related to with our cultivated ones. So we have used several barcode genes under the phylogenetic approach, in which my colleagues in Delhi National Bureau of Plant Genetic Resources have helped us, because we had the complete responsibility of cytogenetics. So they helped us in sequencing these genes and providing the data. And at native, we have concentrated on these 4 genes, and based on that, we have developed the phylogenetic relationships. And this is the list of genes which have been actually suggested, and we have taken almost all of them, distributed among the working partners. And the first aspect I would be talking is cytogenetics. We have done the detailed morphology of several species. These are the ones which are cultivated, and so as you can see, there were variability with regard to heteromorphic pairs. There were some polar chromosomes. There is a variation with regard to that. We could able to come to a clear understanding about the species genome characterization. Then we came across, of all the species, just one natural tetraploid, which is Vigna glabricens, whose phylogeny was not clear, so we tried to establish that through our further studies. Cutting short from the karyomorphological studies, we could able to confirm that there is no numerical variation even in the newly described species also. So the number could be confirmed as 22. So we also worked out the symmetry. We have found a distinct differentiation between the cultivated types and varieties and wild species on the other hand. We could able to see the symmetry of karyotype, and the type of chromosomes that contribute to the symmetry were different from the wild and cultivated species. Okay, and then we have also seen that since there is no numerical variation, perhaps structural changes might have led to the speciation, because the species diversity is very vast. So we could able to pinpoint that we have to look for more of structural alterations rather than numerical changes. Then that's what we have concluded at this point of time, that perhaps the structural mutations, the gene mutations might have contributed for the speciation. And this information has already been published in the nucleus and taxon. Then another interesting thing what we have done is we switched over to intraspecific variation. I have talked about interspecific variation. We found new species, we have found the differences. Then we concentrated on intraspecific variation by selecting 2 species. One is inhabited to hilly regions of northeast India. The other one is in the western India, that is Rajasthan, where I earlier worked. I still have attachment with the region, so we have taken one species from that region and we have compared what is the quantum of intraspecific variation with these 2 species. And for this we have used the karyomorphological studies, and this is V. umbellata, which grows luxuriantly in the hilly regions, right? And there is a variation with regard to the number of metacentrics, submetacentrics, etc. But there were no subphylocentrics in any of these accessions. And we have calculated all the typical cytogenetical parameters to assess the genotypic variation. And these are some of the karyotypes which we have built, where we could able to identify certain heteromorphic pairs and certain secondary constrictions. Compared with the other species, Vigna equinetifolia, which, as you can see, it is a species for extremely arid conditions. And now, what is the quantum of variation there in that? Therefore, because our breeders wanted specific genotypes for the specific areas, because the length and breadth of India is so wide that we cannot recommend only one type of genotype first. So we have done that thing, and this is the some of the studies. What finally we came to this thing is, as compared to comparatively, Vigna umbellata had more stable genomic parameters in comparison with the Vigna equinetifolia, which is shown by asymmetry index of the karyotypes, and also we have seen that complete absence of pterocentrics in V umbellata, whereas good number of them in V equinetifolia. So this type of variations we could see. Within the species also we have seen the variation, and finally we have done we have completed that study. I will come back to molecular approach, where we could not able to do it, how our markers and phylogenetic approach has helped us. Then we have carried out some conventional meiotic studies, where we have generated information about various species. These are some of the which are not highlighted. They are the wild and new species which we have come across in our investigations. And the meiotic analysis has also shown us lot of genomic stability. There was not much of any variation. For example, ring and rod bivalents were very common. There were no multivalent associations, and there were one interesting thing we have recorded was the continuous association of certain bivalents in association with the nucleolus, showing the NOR nature of the chromosomes. Then we have found in some cases, I'll just some B chromosomes also have been recorded, which were later found to be segregating at anaphase first as separate, and the presence of B chromosomes was reported, but we did not come across in most of our cultivated species. Only in the wild species we could able to get it. So finally, from this study we have confirmed the number, and also confirmed that the polyploidy has played no report. There is a lot of genomic homology. There are less number of univalents. That means there is a perfect pairing. So that means the genome, as compared to the other example which I will be talking is Abelmoskes, is a very stable. And then we have done a deep study. I will be rushing through some of these slides, because just to say that it is a fluorophore binding pattern, just to venture into the molecular composition of the genomic DNA. Although this is not a final approach, but it does give some information about ATGC rich regions within the genome. At least it gives some idea to start with, so that we can go for advanced studies. And this is how we have seen variation with regard to the fluorophore binding patterns and DAPI binding patterns in these species. Yes, the point is that from all these studies, what we have shown is equinetifolia, which I have talked earlier, and another closely related species, condolences, they had shown high degree of divergence with the DAPI staining is concerned. So that means these two species have some mechanism which is common when it comes to the speciation is concerned. So we had to look into that, whether these two are common or whether these two are distinct, etc. Therefore, we have approached with the molecular approaches to see that what is the single primer amplification reaction, as I was sharing, which is in fact a combination of several markers, common markers, molecular random, rapids, ISSRs, DAMD. We have adopted certain new type of markers also, where they have given a lot of information. And these are some profiles just to make you see that. And based on that, now what was interesting is, among all these, we could able to see that certain accessions were grouping entirely distinct and separate. They were not getting associated with the Tripura accessions. This is also from the same area, but this was able. Now, this is where our interest lies, and so now this has been identified and suggested to our other group to work more on that, so that they can involve it in breeding experiments. Because there is some unique, distinct characters in that morphology. Time does not permit me to go into all the details, but just we could able to identify, locate among the several accessions. Then the other aspect I was talking is about the chloroplast DNA sequences and their sequencing approaches. These are the 4 gene sequences which we have selected for our phylogenetic studies. We have amplified, sequenced and adopted bioinformatic tools and drawn the phylogenetic relationships with a view to see what we have deduced from our cytogenetical studies, whether they go hand in hand with these information or whether it varies. And that's how what we have seen is that when you see the individual gene sequences and build up a tree, it doesn't give much of this thing. So what we have done is we have concatenated all the 4 sequences available with us, another 10 by our group. So we have combinedly come to the conclusion that I'll just show one thing that, yes, you can see the grouping, which is very common. What our interest lies is here those which they did not fall into these respective groups. So these species, Condolences and these Trinervia, these are the species which we still need to be this. Although we have tried to correlate on the basis of cytogenetic studies, not much could be done. So what I am here concluding is that these are the 3 species which we need to collect more accessions, more materials, and go for detailed study. Then only we can come to a proper conclusion. So this was this thing. So overall conclusions from this system was that mitotic study has told us about the genome, stabilized genome in the absence of telocentrics and subtelocentrics in few, and in wild species the story is different. Then meiotic analysis has confirmed the n basic number as n is equal to 11, and the heterochromatin pattern has informed about the nature of some of the wild species that we were talking about. Then the molecular approaches, they have helped us to identify certain accessions. Just I am mentioning only one here, that is Tripura accession. And similarly, and here, as I said, that Condolences, Hyneana and Phylobenta, now these are being focused because they could able to be isolated. I think I should go right. The second one is Abelmoschus, which is commonly known as okra. Why I selected this plant is, look at the starting point itself, which is very diverse. We had just one chromosome number, 2n is equal to 22 in all the Vigna species. And look at the mosaicism here, 56, 58, 72, 130, 132, 200. The number is very high. So literally there was no information when we started work with which species, although there were some initial efforts, but clear-cut information was not there. Therefore, first of all, we have recorded all the diversity available in the species. Yes, they are not the leaves of the same plant. You can see the pinnae, they are different. 5, 6, 7, 5 and 3. Initially we thought that maybe, but each species has a very distinct diversity with regard to this one, and floral diversity was also seen there. And so what I just want to say is that the Abelmoscus, despite being the fact it belongs to one of the very, very popular family, that is Malvaceae, to which many of the important plants, jute, China rose, coca, etc., cotton, which is a priority plant of Tamu, I know this department, which has, but this species, this genus has not received any important or any attention from the cytogeneticists, because I can tell you that morphology was extensively used. Based on the simple morphology, many of the classifications were done. Of course, there was one attempt of classifying them on the cytogenetics basis, but unfortunately, they were based on not very clear preparations or not very clear this thing. But these were the initial studies. We got richly benefited by these studies because at least we knew that what are the problems there. Therefore, and these are for camera lucida diagrams, they are not this one. So this has helped made us to make a comprehensive effort, taking almost 10 species are available in our Indian subcontinent, and we have taken almost 92 or 93 extractions, and then we have done the extensive studies on that. And 2 species, Palianus, and in fact this is a new species named after the institution NBPGRNCS, National Bureau of Plant Genetic Resources. It has been named after the institution. It is published, so the all this information is published. So then just to mention about the dynamism of the chromosome numbers, I am just showing these slides. You can see 130, 66, 32. So the very first effort was to get a clear cytogenetical understanding of the species, then apply molecular approaches. That was our challenge. We, to a larger extent, I hope that we were able to see that, because despite the high numbers, we could able to, at least the number part. I am not talking of the morphology, although you can see some of them are very clearly able to do that, but we didn't indulge in that. We just restricted ourselves to numbers. So these are different species: 58, 66, 72, 130, 130 again, different species, 132. And you can see that you not just numbers, but in somewhere we can even clearly see the morphology. Some secondary restrictions could also be found out, and ultimately 200 of them. So thing is that because of the problems of cytogenetics, this material was not properly investigated. So we had a confusing view. Which species is closely related to which? What was this? Therefore, our next job was to develop a concept. What we have done is we tried to group them and see that there are group A and group B, which we consider as basal species. And there are some others which have higher chromosome numbers, which are the derived species. And this callate with 200 is a example of a typical amphidiploid. So today we know what is the genomic status of these species, how we can plan our experiments of breeding, how can we improve them, which species needs to be crossed where. That information we could able to get it that. So this is what has been the outcome. And yes, this information also, first we tried to publish, and this information has been published in the Journal of Horticulture Science and Biotechnology. Then meiotic studies, without meiotic studies it could not be complete the story. So then we indulged into that arena where we comparatively got good success, where we could able to analyze them. In the basal species there were no multivalent associations. Only in the basal species, group A and group B, which have been, I'll show the classification, scheme of classification we have proposed. And it perfectly works into that scheme. So this is what just I wanted to see. But there are some derived species where we could able to see the quadrivalents, univalents, trivalents, etc. And so these are these things. As you can see here, these are the higher numbers, and you will find the quadrivalents present in this, and more bivalents in a diploid species. More number of bivalents means there is a more genome homology and there are perfect pairing. So that means these are the not any tetra. Despite their higher chromosome number, they are basic and basal species, which have led to the development of other species. So that is what we have concluded. I am not be going into these details. So this is what we have done. We said that these are the species that we are talking, basal species with the 66 and 72 giving rise to 130 and this thing. Now there are some links missing in our study. We are looking for those materials. So this is the scheme that we have proposed. And another aspect is hybrids. Unless you see the behavior of chromosomes in a hybrid environment, we cannot just conclude that this is a diploid or this is a tetraploid. So those things we have completed, and this information has also led to the same type of conclusions. I'll just see that this scheme again has worked perfectly well into this thing because, oh, I'm sorry, this has been. So this is a scheme that from the chromosome numbers we could able to design, and this work has been accepted for publication in Genetic Resources and Crop Evolution, which might be coming up in the next few months of this thing. Okay, so I could have gone for a detailed explanation, but then time is the problem. Then we have done the same type of studies, but this did not offer us much of help here. Then we have switched over to molecular approaches, because wherever we have found some unique types or unique this thing, how they are behaving and how they are this thing. But you will be surprised to know that here nothing much could be deduced from the molecular approaches as much as we could able to come from the cytogenetical studies, because they are just grouping into, each species is grouping into respective things, so you cannot make out anything from these studies. And even molecular phylogeny also did not, I'm just rushing through this because yes. Now this is the important information that we put, important or whatever it is, from the molecular approaches, which we already could able to get from our cytogenetic studies. Therefore, I just would like to make a submission here that we have diversity of plants, we have diversity of genetic structure and genome. What we need to is, to understand it, we have to be selective in our approaches. In some cases, maybe a conventional cytogenetics, which is very, very routine, can help. And in some cases, maybe molecular approaches may not offer you more of answers, but basic cytogenetics can tell you. For example, in case of Ebelmosters, initially it was neglected because the technical problems were there. So we didn't had good microscopes, good lenses, etc. So people have not gone much beyond just looking at some chromosomes. But today we have all these things. So accordingly we can adopt appropriate techniques, and we can come to a meaningful this thing. Since we had some issues which were unable to address by all these things, that is what brings me to here to this department, to Professor Stelly's lab, where I would like to learn more about the fish technique and just once. I'm sorry I could have, yes, this is a small group of is a small group of my students who are working with me with the diverse plants, and she is the one who worked on Vopra, Merita, and she is worked on Vignanju, and others we have worked on cucumis, citrus, and Santosh has completed his work on orchids. So in all these diverse plant materials, we are using this approach. That's all I wanted to say. I'm sorry if I have exceeded my time. Sorry for that.
Good.
Okay. And these are some of the associated scholars in other labs. So thank you very much.
A few questions?
All those species of okra,
Yeah.
are they consumed? Do people, is that the okra I would see
Yeah.
in my garden? People eat the
Yes,
the
yes.
product of all of those species?
Yes.
Not all of them are edible, are they?
They are
All of those
most of
species?
them are edible. Some of them are medicinally important because of the high mucilage content. They use in the folk medicine also. Some of these, if you have a cut, they just rub it. Some of them are not edibles. But Esculentus, Moscatus, subspecies Moscatus, these are the highly edible ones. I think this picture shows
And does
the
does
edible
taste-
items made by.
Does their taste vary?
Pardon? Yeah, they
Does the
taste.
taste from different species?
For those who have a taste for that type of this thing, it definitely is there. That's why Indian government wants to spend more money to get better hybrids in this. And in fact, some of them are even ornamentals, better ornamentals. I have shown you the diversity of their floral morphology. Some of them they want to develop as a horticultural varieties.
So what, with that range of chromosome numbers, and I would presume a lot of other underlying genomic differences, what kinds of problems do breeders encounter in trying to hybridize and backcross and utilize that germplasm?
Yeah, that's what these are not new things, but we have checked up in our system for the first time. For example, whenever we are using the derived species, as I mentioned, Cali, Abelmoschus cali, and Abelmoschus tuberosus, variety tuberosus, whenever we are trying to cross them, there are so many problems. I mean, the F1 hybrids mostly, maybe the high chromosome number, I do not know. I mean, the breeders could not get success. I am not a professional plant breeder. My colleagues are there. They are doing. That is number one problem. Number two, even if you try to cross and diploidize them to stabilize through colchicine, problem is there is no seed set. Despite doubling, there is no seed set. So we have to try for some vegetative propagation methods for such, especially the ornamentals ones.
So you say one of the species is tuberosum? Does it form
tuberosus.
It forms tubers? Is
It does form, but the plant is more known for the fruits, not for the tubers.
Right. So is that the one that has high univalence?
Yeah,
So it's sterile?
sterile, that is the.
So it's propagated vegetatively?
It is done by tubers, but problem is when you want to go for crossing, your
Sure.
seeds need to be this thing there.
I didn't even know there were different varying oaks. So it's kind of, for me, it's kind of interesting to hear this story because Gossypium cotton went through a lot of these same things about 1930. 1940 was when the genomic relationships amongst the different species and the ploidy relationships were ironed out. So right around 1940 was when that was first published, and so of course we've been using that information for 80 years now, or 70 years now. So it's interesting to see them wrestling with the same questions that were dealt with a long time ago here, because they do give you once you have that information, which you obviously have some advantages because you have molecular techniques to combine with the cytogenetic ones. But it's on that foundation then that you can really move forward. So, any other questions? Yeah.
So how many okra breeders are there?
Not, at least I can vouch it for India. We do not have many of them. In India, I know there is one agricultural research station in the southern part of India. Another problem with the apple moschus is some of the distribution, right? All of them do not flower at one place. So you have to move zigzagging the country, which requires a lot of travel funds, and that's big. So there are not many of them. I can precisely say at the most 3-4 of them are doing this job.
Okay. So moving to breeding from wild species is always kind of a pain. So what are there any problems? The reason why they would want to go to the wild species rather than just work with the domesticate?
Definitely. Otherwise, what is the use of these wild species? Unless you bring them into the breeding program and unless you make use of them, there is no point in just crossing the species. You maybe just inbreeding would be there between the species, but unless wild species are brought into this program, which my colleagues are doing now.
Well, what's the problem, the reason why they would bring the wild
I
species?
just mentioned to him, the problem is that flowering. Flowering is one problem because these wild species, their flowering season is different. The diploid species.
No, that's the problem to bring the wild species in. Why would a breeder want to bring wild species in? Are there diseases? Are there yield? What's the advantage of working with the wild species in breeding?
Okay. I do not have a straight answer for you. I'm sorry for that. But then I still speculate. Our information about wild species and within the wild species there is a lot of diversity. Now, which one you are catching and which one you are concentrating to cross with that? First of all, you have to see the broad base. That is also not done. I was just sharing because for me, as cytogeneticist, the number was a challenge, so we could able to create. I started like that, but then now I am deep into the business. I understand that we have to do a lot. This is just a part of my study. Yeah.
Can you guess how many of the species are actually cultivated for either food or other medicinal purposes? Right. And so the breeder is going to be, let's say, breeding for food purposes, or they're going to be breeding for medicinal purposes.
Okay.
Do you have an idea how many species they would be dealing with?
The Esculentus is one species which is basically adopted all over India, number one. Number two, preferred by most of the people all the parts of India. And there is another one, this Calais, Abelmoschus Calais, which has a very large fruit size, okay, and a very better taste also. Better taste, not bitter taste. So we would like to propagate these two species more and see how the breeders and farmers, how they respond to it. It is there, but better varieties are lacking. That is the problem. And we don't have anything to offer off the counter that these are the better, because information is incomplete.
Do you think the breeders would be most apt to use the other species as sources of disease resistance or
one is the
other characteristics?
Yes, they definitely have to do it. There is a long way to go. As I shared with you, I started because of my love for the chromosomes, but I ended up with dealing with a very challenging system from all, not only cytogenetics. In the breeding is a very complicated one. Yeah. But we are trying. We are into the business and we are doing. We will come out to it.
Very good. Okay. We're at the 1 o'clock limit, so join me in thanking Dr.
If you're sitting in a red or